our partners at Akron Children's Hospital Vision Center, Cleveland Clinic Cole Eye Institute, and University Hospitals Eye Institute. We are so proud to support these world-class institutions through our grant funding aimed at improving strategies to prevent, treat, and reverse vision loss that's associated with eye disease in children and adults. For our constituents who are attending today, welcome, and thank you so much for your investment in the Cleveland Eye Bank Foundation. Funding from individuals, corporations, and foundations have allowed us to support vision research and education since our very beginning as the Central Eye Bank for Sight Restoration back in 1958. We hope you are inspired today with your gifts that have impacted vision here in Greater Cleveland, Northeast Ohio, and beyond. Thank you.
Welcome. I'm Alan Roth, President of the Board of Directors at the Cleveland Eye Bank Foundation and an ophthalmologist and cornea surgeon at Cleveland Clinic's Cole Eye Institute. On behalf of the Board of Directors, we are proud to support and bring to you today new discoveries that will have an impact in combating debilitating eye disease in the future. I see firsthand as a practicing ophthalmologist when new procedures, new techniques, and new treatments can improve the vision of my patients through new interventions. We appreciate your attendance today, your interest and support that helps give scientists the tools they need to discover new therapies and cures for vision loss. Enjoy the program.
Thank you, Alan and Debbie. Good afternoon. I'm Jonathan Lass, and I'll be the moderator for this symposium. Also a member of the Board of Directors at the Cleveland Eye Bank Foundation and a practicing ophthalmologist and researcher at University Hospitals Eye Institute and Case Western Reserve University, I'm pleased to moderate this symposium presenting the diverse and impactful research from physicians and scientists supported by the Cleveland Eye Bank Foundation, whose work spans vision issues in children to age-related eye disease in adults, including corneal infections, glaucoma, diabetic retinopathy, and macular degeneration. Before we get started, I wish to take a few moments to explain some of the logistics of this webinar. Your audio lines are muted to eliminating background noise at your location. We're going to welcome 2 minutes of questions after each of our first three presenters. And the way you'll submit those questions will be through the Q&A function on your webinar. After the three presenters, we're going to hold the questions for presenters 4, 5, and 6 and conduct a mini panel discussion with the questions after speaker 6. This is in the area of retinal degeneration. And then we're going to have presenters 7, 8, and 9 conduct another mini panel discussion with questions for those speakers. This is in the area of diabetic retinopathy and imaging. Please, as I said, please feel free to send your questions through the Q&A function. I'll be monitoring that, or the chat function, and we'll try to handle your questions either after the speaker or we'll also have a Q&A at the end of the program for all the speakers. I'm going to let the speakers know 1 minute before their 8 minutes is up that they have that 1 more minute to wrap up. And finally, a reminder to our speakers to unshare your slides at the end of your time so the next presenter can start their talk. So let's get started. Our first speaker is Richard Hurdle, who's the Chief of Pediatric Ophthalmology at Akron Children's. His talk will be on new standards for treating visual system diseases in infants and children, the impact of decades of professional, patient, and community cooperation. Rich?
Jonathan, thank you very much. I'd like to start out making sure that you can hear my voice and see my slides. It looks like everything's working on my end. Just want to make sure it's okay. And say how grateful I am that the Cleveland Eye Bank Foundation has asked that we represent Children's Hospital and the Vision Center in the work that we're doing to try and improve the quality of vision in children. It's an honor to be able to participate in this panel. Over the next few minutes, I'd like to highlight some of the most important discoveries and new treatments in the world of pediatric ophthalmology and vision science. And this will go over a bunch of material, but all of this is published in thousands of pages of literature. So I'm just going to highlight very quickly what's happened over the last 15 to 20 years, specifically as the result of community involvement and participation and the absolute need for external funding to make this kind of science possible. But I wanted to start on the same page because I don't know where the audience is coming from, what levels the audience are at, what we understand or don't understand about science. So I wanted to kind of get us together a little bit and just come up with a couple of definitions that I use that I think most of us that do science are familiar with. And I apologize in advance if this is rudimentary for some of you who are watching, but the scientific method is something that we use whether we're doing work in a laboratory or with patients. And this has been around actually since about 500 BC, but it hasn't been formalized for until the last 100 years or so. With the advent of the human protections that have been put in over the last 50 to 70 years, we now have modern clinical science and structure to our clinical trials, with the height of these being the randomized clinical trial. This is a typical pyramid in your upper right showing the levels of evidence that we use to transfer care or information or new knowledge directly to patients. So it starts out with ideas and research in animals and sometimes on single cells and plates and laboratories, and goes all the way up through single case reports and case series. And then multiple trials are done where the patients are given drug or placebo or treatment or placebo. And then these are multiple centers using multiple populations of patients are published, and then multiple studies are put together. And as these levels of evidence increase, the confidence that the clinician feels with prescribing that particular treatment becomes enhanced. And this is the method that most of us use who do clinical science. So I'm going to talk a little bit about the trials that have used this. Retinopathy of prematurity is still around. It's still a blinding disorder, and it's the result of being born too early. Although oxygen gets blamed, it's not primarily responsible. It's the primary responsibility is the baby is born too early. And all of the factors that go into the prematurity result in the blood vessels becoming abnormal in the developing eye. Even with treatment now, and this is with treatment down on the bottom, about 500 babies still go blind every year. But before this, thousands of babies went blind every year. And this is the result of National Institutes of Health supported clinical trials. And these are the trials that you see listed up here: the cryo-ROP, the stop-ROP, light-ROP, and ET-ROP. And these are trials that involve thousands of patients at tens or dozens of institutions since the mid-'80s up until recently. And what these studies found was that cryo, or freezing of the eye, decreased the incidence of blindness by 50% in infancy and 17% by age 10. Supplemental therapeutic oxygen delivery was a hypothesis that giving oxygen at a certain time during the development of ROP could decrease its severity, and that didn't show to be positive. Also, there was a hypothesis that light was a problem, especially during certain periods of development of the retina in the neonatal intensive care unit, and there was no decrease in ROP with light reduction. The early treatment of ROP study with laser showed that doing early treatment reduced the blindness another 5%. And the most recent trials showing artificial intelligence, the use of telemedicine, have shown to increase or decrease blindness worldwide by the result of showing photos to specialists all around the world. And now we also use intraocular Avastin. And there are the trials that have been done comparing laser to Avastin are showing equal efficacy with equal long-term benefits and less side effects to using intraocular Avastin. The PETA group was started in 1997 And its goal was to study pediatric eye diseases, specifically ones that are commonly treated by practicing pediatric ophthalmologists. Since it's been funded then in 1997, you see that there's sites in 3 countries, and hundreds of ophthalmologists and optometrists have participated in these trials. It's been funded with over $65 million, and you can see all the protocols that have been done and have been completed as a result of this funding in 3 countries. And it's involved 10,000 patients in all of these cities and has resulted in 150 publications. These are some of the major findings that are actually were not standard of care prior to these trials. For instance, patching 2 hours works as good as patching at 6 hours or 8 hours in moderate amblyopia. Patching 6 hours works as good as full-time in dense amblyopia. Using atropine can be as effective as patching. Using glasses alone can treat amblyopia, even without patching or glasses or drops in strabismic amblyopia. And the changes in the current standards of practice in common eye diseases, such as strabismus and amblyopia, are the result of these large-scale clinical trials. So we're moving from anecdote. My mentors taught me how to treat amblyopia this way, so this is the way that I treat it. I was taught how to patch this way, so this is the way I'm patching. These clinical trials are now showing us rigorous ways to treat amblyopia and strabismus. Another really interesting trial was the hypothesis that using an intraocular lens in the first 6 months of life in a child with a unilateral congenital cataract would be better, the vision would be better than using a contact lens.
Sorry to interrupt, 1 minute left.
Okay, so the children were randomized to either of these 2, and it was found that the vision was equal in both of these, with the contact lens or the intraocular lens. And this was the main findings. The last group of trials were convergence insufficiency, and that's a disease that's present in about 5 to 7% of people. And this was done largely optometric based, and it showed that doing therapy in the office with home reinforcement was better than any other type of therapy, including pencil push-ups. And we did another convergence trial, which showed that convergence therapy used to try and treat attention and reading did not help contention and reading, but only helped convergence. So what we found in these thousands of people with these multitude of studies is that it really was the support of the community by allowing their families and their children to participate in these trials, and the grants, endowments, and gifts allowed us to progress in pediatric eye care. Thanks for listening.
Thanks for Rich. We don't have any questions from the audience. I have a question. You didn't touch on this, but there was already a movement towards telemedicine in managing many of these diseases. And obviously COVID has increased that activity. Could you comment on the use of telemedicine and where that's headed? Artificial intelligence plans for, let's say, monitoring ROP, et cetera?
Yeah, I think telemedicine has right now a limited place in pediatric ophthalmology because it's difficult for us to get the information via online routes. Photographic documentation and interpretation of photographs using artificial intelligence for known eye diseases, I think, is a big opportunity, equivalent to diabetic retinopathy. So diseases like retinopathy of prematurity probably will have the largest impact in telemedicine, where we can actually quantitate the disease. There are now algorithms to quantitate the severity of ROP with standard fundus images similar to diabetic retinopathy. So I think that's our best in right now.
And what is we have an interest in using atropine for the treatment of myopia. What is your feeling? I noticed Pedig has studied that. Mm-hmm. Should all our kids who are getting nearsighted get on atropine? So
there's definitely a proven benefit of using low dose atropine for reducing the progression of myopia. Unfortunately, it's like some of the glaucoma studies, the amount of myopia that's reduced is not significant. We're talking quarters to halves to three quarters of a diopter. There's no studies that have shown that using myopia goes from pathologic to non-pathologic myopia. So I think the utility of it has yet to be shown clinically.
Okay, thanks very much, Rich.
Sure,
Okay,
thank
we're gonna, I noticed there was other questions, we'll handle them. We're gonna move on to our next speaker, who's Connie Tam, who's a basic scientist at the Cleveland Clinic Cole Eye Institute and Learner Research Institute. She'll be speaking on harnessing the dual functions of CAMPs to treat corneal infections and inflammation. Connie?
Hi, everyone. I am so excited here to show you the new discovery in my lab about CAMs being a potential treatment for corneal infection and inflammation. So just a month ago, I read a story about a 2-year-old boy in Chicago, and he almost lost his vision after contracting bacterial eye infection. His eye turned really bad just within hours. It was fortunate that the infection was discovered early on and it was responsive to antibiotics. So the boy made a full recovery. The story really emphasized the importance of effective antimicrobial therapy to a good visual outcome. That said, we are all facing serious challenges that are reducing the effectiveness of classic antibiotics. The top challenge is antibiotic resistance. The two most common ocular pathogens are Pseudomonas aeruginosa and Staph aureus. These two are high priority bacteria that require new antibiotics. The resistance issue won't go away until new drugs are available for clinical use. This brings up the second challenge, which is insufficient antibiotic pipeline. The major issue is that the relatively new drugs are mostly derivatives of existing ones that the bacteria are already resistant to. So we do need to speed up the time innovative drugs are made available in the clinic. And the third challenge is limited choices of ophthalmic anti-inflammatories. The use of topical steroids could be beneficial in some cases, but the risk for side effects and complications are still concerning. One of the research areas in my lab is anti-infective and anti-inflammatory drug discovery. So the small molecules that we've been studying are CAMs, which stands for keratin-derived antimicrobial peptides. They are small fragments belonging to the keratin 6A protein. And this protein is one of the building blocks that form a filament network in epithelial cells to maintain cell structure. We found that small fragments with specific amino acids coming from the K6A protein can kill bacteria. Here you can see that under the microscope, the bacteria in the culture media of corneal epithelial cells are inactivated after CAMs are added. So using transmission and scanning electron microscopy, we can see better what's happening to the bacteria. Here's what normal Pseudomonas looks like and versus those treated with CAMs. It's clear that CAMs can make severe damages to the bacterial cell membrane. So we also see the same kind of structural damages in Staph aureus treated with CAMs. As we are interested in anti-inflammatories, we test whether CAMs can reduce inflammation by regulating production of cytokines, the important mediators of inflammatory responses. We collect mouse neutrophil and macrophage, and then we put them in the wells of cell culture plates and stimulate them with LPS or LTA, the inflammatory bacterial components. In this assay, some samples are given CAMs before the stimulation and some receive CAMs after the stimulation. And after 8 hours, we collect the cell culture media and measure how much cytokines the cells have produced. Here I'm showing 2 of the 6 cytokines we measure as representatives. When neutrophil and macrophage are stimulated with LPS or LTA, cytokines and chemokines are produced. What we found is that adding CAMs to the cells before or after stimulation demonstrate dose-dependent suppression of cytokine production. And meanwhile, using control peptides with a scrambled sequence of CAMs cannot suppress any of these cytokines. And to test whether CAMs work in the eyes, we need to move forward to the mouse models. But before that, let's check and see if CAMs are non-toxic. So we incubate human corneal epithelial cells or repa cells with CAMs and then measure the viability of cells. For comparison purpose, we include samples that are treated with BAK, a common preservative that's known for its toxicity. Here, we don't see any toxic effects from CAMs when compared to the untreated controls. As we put CAMs or BAK on the mouse eyes and then check the eyes with fluorescein, we don't see any epithelial defects or damage with CAMs, whereas the corneal surface with added BAK is damaged. So overall, CAMs appear to be safe on the eyes. Now we're ready to test the peptides in the mouse models of corneal diseases. The first one is non-infectious inflammation. So we induce corneal inflammation with dead bacteria, Pseudomonas or Staph aureus, and give CAM eye drops either before or after the induction. 24 hours later, we measure the levels of cytokines and immune cells in the inflamed cornea. Here I'm showing one cytokine and the number of leukocytes, which include neutrophil and macrophage, but they share the same trend across the board. As we've seen before in vitro experiment, CAM eye drops reduce inflammation in vivo by suppressing cytokine production and immune cells infiltration. Now we come to the corneal infection. Now we come to the wound healing model. So this model, we use it to see if CAMs can have any effect to the epithelial cell closure, wound closure. So in this model, we see that while suppressing inflammation, CAMs still accelerate epithelial wound closure as compared to the group received the control peptide. Now let's come to the corneal infection model. Mouse corneas are infected with either Pseudomonas or Staph aureus. And to look at disease prevention, one group has no treatment and the other one is given topical CAMs at the time of bacterial inoculation. And the pictures here show you that what happened after 24 or 48 hours after we put the bacteria on the eyes. And you can see that CAMs eliminate the bacteria and protect the eyes from getting infected. And to look at the therapeutic effect, mouse corneas are infected with either Pseudomonas or Staph aureus, and we wait 24 hours before we give them CAMs. Compared to the group with no treatment, the red ones, and they have disease progressing, the mice receive CAM eye drops have the infection controlled as the overall presentation of their eyes is getting much better within 1 or 2 days after treatment. And the bacterial load and immune cell infiltration are also reduced. So just a quick recap of the key points about this work.
1 minute left.
Antibiotic resistance is a pressing global issue and is posing a serious risk for vision loss caused by ocular infections. Therefore, actions to invigorate development of novel antibiotics are critical. My lab used a steroid inflammation model to show that keratin-derived antimicrobial peptide CAMs have anti-inflammatory properties which are independent from their bacterial killing action. And in a corneal wound healing model, CAMs accelerates epithelial wound closure to restore the protective barrier of cornea. In the corneal infection model, CAMs can block infection and reduce inflammation at the same time to improve disease outcome. So with that, thank you to all the members of my lab and the core facilities at the clinic and Case Western for the great efforts put into this work. It's made possible by the generous support of the NEI, Eversight Center for Eye and Vision Research to Prevent Blindness, and the Cleveland Eye Bank Foundation. Thank you.
Thanks, Connie. No questions from the audience. Please remember to use that Q&A as things come up. Connie, infections are certainly occur with contact lenses, and patients can present with different pictures with inflammation. Do you see if someone with a contact lens wearer presents with a early infiltrate, central, peripheral, how at one point do you think the CAMs might be utilized with a culture, without a culture, etc.?
Yeah, so based on the in vitro experiments, we noticed that CAMs can kill different kind of bacteria, gram-positive, gram-negative. And the one infection model I just showed you to look at therapeutic effect, we actually put the CAMs onto the eyes after we've seen opacity on the mouse eyes. So like you said, if you are in the clinic and see patients already developed opacity after contact lens wear, I think like it's reasonable. Like I'm just making a guess that in the future when we have developed it to a more mature state, including improving the formulation, it is okay. I think it would have good effect on the disease outcome.
So you see this using this as an early event in terms of instituting therapy early on before it advances.
Definitely, the earlier you put on it, the better. But we are also looking at the synergistic effect, if there's any, between the CAMs and the known antibiotics. Perhaps, if disease has already progressed to a more severe state, we could have CAMs added to known antibiotics, just to minimize the resistance to develop. And hopefully we can get better therapeutic effect that way.
All right, great. Thank you. Very exciting. Okay, our next speaker is going to be Doug Ree, who's a professor and chairman at Case Western Reserve University, ophthalmology and visual science. He's also director of the UHI, University Hospitals Eye Institute. He's going to be talking about understanding the drain of the eye to develop new treatments for glaucoma. Doug?
Hi, good afternoon, everyone, and thank you for inviting me. And again, thank you to the Cleveland Eye Bank Foundation and its board members and donors for its support. Let me go ahead and share my screen. Okay, and let me go to, all right, very good. So I'm going to be talking about understanding the drain of the eye to develop new treatments for glaucoma. These are my disclosures. I don't think any of them are, these are my corporate disclosures. I don't think any of them are relevant here. I'd like to give a brief introduction about what is glaucoma, the normal and abnormal control of eye pressure, the public health implications of glaucoma, and the clinical progression, as well as talk about trabecular meshwork research. So glaucoma is a condition in which the pressure inside the eye is too high for the optic nerve to withstand. You can think of the pressure being created by the fluid flow, the fluid being produced by the ciliary body and then flowing through the pupil and going through the drain of the eye, which is the trabecular meshwork. What goes wrong in glaucoma, what the pathophysiology is the an altered drain. So the drain is kind of blocked, and you can see it statically in this diagram here, but you can think of it conceptually as the fluid trying to circulate around, and then that increased pressure squashing the optic nerve. So the problem again is with the drain of the eye. Glaucoma is a major public health issue. It affects 2% of all Americans, affecting all age groups from newborns up to elders. It's the most common cause of legal blindness in Americans of African or Latin descent. It is the second most common cause of legal blindness in Americans of European ancestry. In persons over the age of 60, it's relatively common. In persons of European ancestry, it's 1 out of 20, 5%, and out of African ancestry, 1 out of 10. So it's pretty common. And with Latin or Asian ancestry, it's about 7%. Clinically, this is what the spectrum looks like when we look at the optic nerve and test the visual field to see what areas the patient is no longer able to see. And then if you were to, this is an artist's rendition of what the vision loss would look like. And it starts from the outside, kind of moves in. And the cruel aspect of glaucoma is that it can actually take everything away and make, essentially darkness, completely like midnight. So I'd like to now move into trabecular meshwork research. So why study the drain? How does the drain work? And just to acknowledge or show that extracellular matrix, and I'll explain what that is very important. So we know that disease, and I showed you clinically that the pressure is up and it shows at the optic nerve. So the clinically, the disease is at the meshwork and also at the, I said clinically, I meant anatomically, the disease is in the meshwork and in the optic nerve. So if you were to focus in on this area and blow it up, we're looking at the juxtacanalicular region, which is this area right next to Schlemm's canal in which the resistance is regulated. So this is the area, the anatomic location that controls resistance. What do we know about this area? It is not a static structure. It is constantly undergoing remodeling. So this is a schematic of that same area, just flipped over a little bit down. And this is, again, the area that I'm talking about. And if you were to blow this up or magnify it as a schematic, there's two main pathways here. There's the pathway in between cells, which is called the paracellular pathway, and this pathway through cells called the transcellular pathway. Regardless of which way the fluid tries to make it out of the eye, this orange and black substance is called the extracellular matrix. It is the ground substance on which the cells sort of sit and find their structure. And you can think of it like mortar in a wall of bricks. So the matrix, the fluid has to get through the matrix, and the matrix ends up being very, very important to controlling the eye pressure. We do know that the only known pathophysiology for glaucoma, for primary open-angle glaucoma, is an increase in the amount of matrix that is found in this juxtacanalicular region. And a schematic, this is from Johannes Rowan's original paper describing this in the early 80s, where you can think of this matrix as sort of constipating the flow of fluid trying to get out through the juxtacanalicular region. Despite the fact that we have a myriad of medications, a myriad of different surgical procedures, sort of a dizzying array, but all of them do one thing. They lower the intraocular pressure, and they are palliative. They do not lower the pressure by altering the disease pathophysiology. They lower the pressure by altering things that are pathways that are normal to the eye, but they don't actually interrupt the pathway, the disease pathway. So what that means is it's palliative, and the disease, even though we lower it, can still be progressing. Even though you don't necessarily get visual field loss, but we constantly have to see our patients to make sure that the pressure is controlled, and we might need to advance treatment if it's not controlled. So despite the best of care, because we're not directly intervening in the disease pathophysiology, what's called disease modifying therapy, about 7 to 14% of patients will still go legally blind from glaucoma in one eye over a 20-year period, even with the best of care. So one of the things I'd like to then turn our attention to is a protein called SPARC. Before I do so, I need to tell you a little bit about this cytokine growth factor called transforming growth factor beta 2. This cytokine is elevated up to 3-fold, so it's found in much higher levels in the aqueous humor, the fluid in the front part of the eye, in patients with open-angle glaucoma. And if you use TGF-beta2 in experimental model systems in human anterior segments, you get an increase of intraocular pressure, and you get an alteration of that juxtacanalicular extracellular matrix that resembles glaucoma. And if you use it in a live system, so in mice and rats, TGF-beta2 increases intraocular pressure. You may not suspect it, but mice are incredibly good model systems. Outside of non-human primates, mice have the most similar aqueous drainage system to human beings.
Sorry to interrupt, 1 minute.
Got it. Our hypothesis is that anything that controls extracellular matrix would be relevant here. Matricellular proteins allow cells to control their surrounding extracellular matrix. We examined the family of matricellular proteins and found that 3 of them actually do lower intraocular pressure in these mice if you take out that gene. And so with SPARC, we know that it is, I just want to highlight across the street, they found, John Crabb's lab, that found that SPARC is the most highly secreted protein in response to TGF-beta2. So people had shown that TGF-beta2 increases SPARC and increases IOP. Our hypothesis was that it was dependent on SPARC, There's this big magic box that we spent the last 10 years trying to elucidate. I'm not going to go into the details, but after about 10 years, we found that we started to uncover what the pathways were. But our most important finding is that if you give TGF-beta2 in normal mice, you get an elevation of intraocular pressure, which you would expect, and others have shown. But in mice that are not able to secrete spark, if you take out the gene spark, TGF-beta2 cannot increase the intraocular pressure. This ends up being highly, highly important, kind of the holy grail, because mice that can't make spark can't get glaucoma. So as far as what we're doing now and some of the work that the Cleveland Eye Bank Foundation is helping us elucidate is what are the different pathways that make up those two other arrows. It probably looks something like this. I'm not going to bore you with the details. Here's a cleaned up view, but we are starting to uncover these pathways with the hopes of interrupting them and in fact interrupting the disease pathophysiology. So this is one of the few proteins that's been shown to regulate intraocular pressure. Bottom line, we've discovered a protein that regulates eye pressure normally and is likely critical to the pathophysiology of glaucoma. I'd like to thank my collaborators and in particular the Cleveland Eye Bank Foundation for their support of this work. Thanks, John. Sorry for running over.
Doug, one quick question. How do you think this will fit in the armamentarium of various medical treatments for glaucoma, let's say compared to the prostaglandin analogs, in terms of potency, in terms of ability to lower intraocular pressure?
So interestingly, it probably won't necessarily be that potent in terms of lowering pressure because we only found about a 20%. But what we hope it will mean is that once the patient's on this treatment, they won't need additional treatments once they're controlled. So it won't require the same amount of follow-up. You'll actually have halted the disease process at that point and essentially save their vision.
Thank you. Well, we look forward to continued developments in that, and hopefully there's a new class of agents for glaucoma treatment. Thank you. Okay, well, we're going into the next phase of our seminar where we'll be focusing on the retinal degenerations. And our first speaker will be Paul Park, who's an associate professor in our Department of Ophthalmology at Case Western and University Hospitals. And he'll be talking about improper protein production in retinitis pigmentosa. Paul?
All right, thanks, John. And I'd like to thank the Cleveland Eye Bank Foundation for putting together this symposium and giving me a chance to briefly share some of the work that we're doing in our laboratory. So as you're seeing, and as you will see in the presentations in this symposium, when we're talking about eye research or understanding normal physiology, we could look at things at various levels, all the way from the patient down to the organ level, tissue level, and all the way down to cellular and molecular levels where you're looking at individual proteins as well as small molecules. And in our laboratory, we're basically down to the molecular level where we look at the behaviors and structures and biophysics of proteins. And in particular, our laboratory is interested in the processes that are occurring in the retina, which is here in the back of the eye, and it's a tissue. It's a paper-thin tissue that houses all the cells that are responsible for detecting photons of light and processing and packaging that information for the brain to interpret. And we're interested in photoreceptor cells, which sit in the outermost part of the retina. And these are the cells that are responsible for actually detecting photons of light. And this is just a microscopy image of photoreceptor cells and a cartoon representation. And dysfunctions in rod photoreceptor cells could cause various dysfunctions. Mild dysfunctions include night blindness, all the way to more severe dysfunctions that could cause retinal degeneration and complete blindness. The outer portion of these photoreceptor cells houses the proteins that are responsible for the initial detection of photons of light. And the initial photon detection occurs by a protein called rhodopsin here, which initiates a set of biochemical reactions termed phototransduction. And this is really our the initial events of vision. And to better understand dysfunctions of rod associated with rod photoreceptor cells, we really need a proper mechanistic understanding of these processes, as well as understanding the factors that keep a photoreceptor cell healthy. And that's what the focus of our laboratory is. And we focus on the protein, as I indicated, called rhodopsin. And this is the light receptor. It detects photons of light. And this is mediated by a vitamin A derivative that's conjugated to the protein. And rhodopsin plays a central role in phototransduction as the detector of light, and also in maintaining photoreceptor cell health. And dysfunctions in rhodopsin can cause a range of retinal disorders, including retinitis pigmentosa, congenital night blindness, and Leber congenital amaurosis. Also, photoreceptor cell and rhodopsin activity can also have secondary effects in other diseases such as diabetic retinopathy and age-related macular degeneration. So like any protein in the cell, rhodopsin must be produced properly. And photoreceptor cells have a unique structure where the site of synthesis is in this region here, and the site of action of rhodopsin is in this region here. So like any protein, when the cell synthesizes the polypeptide chain, the protein must adopt the proper three-dimensional conformation. And in the case of rhodopsin, these complex with each other to form higher order structures. And once a proper structure is adopted, rhodopsin is transported from the inner part of the photoreceptor cells to this outer portion where it's incorporated in these discs, which are pancake-like membrane structures. And within these discs, rhodopsin arranges into an exquisite supramolecular organization. And part of our lab is dedicated to understanding how rhodopsin is organized within these discs shown here. And we use a biophysical approach called atomic force microscopy to visualize the organization of rhodopsin shown by these red dots here in real life. And how we do this in AFM, atomic force microscopy, of an ultra sharp probe, which scans the surface and gives you a topographical image of your sample. And this is an image of a disc from photoreceptor cells. And you can clearly see these little islands color-coded here, which represent these rhodopsin clusters. And I'm not going to really talk about this, but I just want to present some of the cutting-edge biophysical approaches we have available in our laboratory. What I will be focusing on in this talk is what happens when the protein cannot adopt the proper structure, but instead misfolds and causes these toxic complexes called aggregates. And when this happens with rhodopsin, this causes retinitis pigmentosa, which will be the focus of this talk. So retinitis pigmentosa is a group of inherited diseases that causes progressive retinal degeneration. It first begins with the degeneration of rod photoreceptor cells. So you get night blindness as well as vision problems in the periphery. And eventually, as the disease progresses, you lose your central vision shown here because of the loss of cone photoreceptor cells. And there's over 40 genes that have been identified that cause the disease, and among them is rhodopsin. So mutations in the rhodopsin gene are the leading cause of autosomal dominant retinitis pigmentosa. There's over 100 mutations that have been identified in patients with retinitis pigmentosa, and over half of those cause the protein to misfold and aggregate. And the mutated residues are shown here in the different colors on the secondary structure of rhodopsin. And it's unclear how these changes in these single amino acid residues cause the protein to misfold and aggregate to lead to a healthy retina looking like this to become messed up, like what this is showing here. And aggregation and misfolding of proteins is not unique to rhodopsin and retinal disorders. You see this across a variety of diseases that you may be familiar with. So Alzheimer's disease is caused by the misfolding aggregation of amyloid beta peptide proteins. Parkinson's disease is caused by the aggregation of alpha-synuclein, and prion disease is caused by the aggregation of prion proteins. And in all of these diseases, great effort is being put into understanding the characteristics of these toxic aggregates that are formed, and also to formulate strategies to disrupt these aggregates to combat these diseases. So the challenge that we have is how do we study these aggregates of rhodopsin? And to do so, we've had to turn to an artificial system that we've developed in the laboratory where we could manipulate
1 minute.
rhodopsin DNA to genetically engineer a fluorescent protein that we could fuse to receptor itself. And we could express these in cultured cells, and then we could apply our biophysical method. And what this method allows us to do, called Forster resonance energy transfer, or FRET, it allows us to determine whether two protein molecules are far apart from each other or whether they are coming together and forming complexes. And we do a further trick here to explicitly look at aggregates by treating our cells with detergents, which disrupts complexes formed by normal rhodopsins, but cannot disrupt aggregated rhodopsin mutants, which we are interested in. And we could differentiate these from our outputs here. So this slide, I'm not meaning for anyone to understand the details of, but what we're doing is we're using and applying this biophysical method on a range of different mutations in rhodopsin that cause retinized pigmentosa, including the P23H mutation, which is the most common mutation detected in patients in the United States. And we're characterizing the aggregation properties of these mutants, as well as we're testing different pharmacological approaches that have been proposed. And in addition to our biophysical approaches, we're also looking by light fluorescence microscopy to look at the cellular biology of these mutations as well. So what we're learning so far from this approach is that there's variability in the severity of misfolding and aggregation, depending on the specific mutation in rhodopsin. As I mentioned before, these mutations cause an autosomal dominant form of the disease. And we have shown that physical interactions between mutants and the normal receptors are not a cause of this autosomal dominant phenotype. And this will instruct strategies on how to approach therapeutics. As I mentioned, we've tested a couple pharmacological agents that have been proposed as a therapy in patients harboring these mutations in rhodopsin. And we've shown that actually a couple of these are probably going to be ineffective, or they may even be detrimental for certain mutations. So we believe that these therapeutic agents should not be pursued any further. And as you'll see in the talks, and as you've already seen in some of these talks, we use animal models to understand these diseases. And what we've shown is that the species background of these mutations have a significant effect, and they can affect the actual aggregation properties of rhodopsin, as well as the effect that pharmacological therapeutic agents can have. So this raises a caution to those studying in animal models to make sure that the animal models and the species of protein that you're looking at actually mimics the human condition if you're looking at things in terms of therapy. So for the next steps, we want to correlate our biophysical findings with studies in animal models, and we have a couple animal models in the laboratory that we're currently looking at. As well, we're trying to formulate strategies to disrupt these aggregates, which we could then test by biophysics and then in animal models. And just to acknowledge the people that have done this work that I presented, Megan and Sri Lakshmi, as well as the different funding agencies, including the Cleveland Eye Bank Foundation, as well as the core facilities we have here in at Case Western Reserve University, UH. So thank you very much for listening.
Thank you, Paul. Unfortunately, we're beyond the 10 minutes. So there are a couple questions, and we'll have to save those to the end. Okay, we're going to. I'm sorry. I'm sorry. I'm confused. No, we'll be able to handle that during the after the other speakers, after Irina and Brian. Okay, so our next speaker will be Brian Perkins, who's an associate staff member at the Cleveland Clinical Eye Institute, and he'll be discussing regenerating photoreceptors in fish models of retinal disease. Brian?
So thank you very much, and thanks to the organizers for giving me an opportunity to speak today. Over the next few minutes, I want to highlight some of our ongoing research efforts to restore vision through regeneration. So inherited retinal dystrophies are genetic forms of blindness that result in degeneration of the photoreceptors, the rods and the cones. What typically happens is that the rod photoreceptors in the periphery of the eye degenerate, and this degeneration progresses towards the center until many people only have a small island of cones left in the remaining foveal region of the retina. Eventually, even these cone photoreceptors die and people will lose all of their vision. Now, the speed that people lose this vision can vary. Some diseases like retinitis pigmentosa may not start until adulthood, whereas diseases like Leber's congenital amaurosis, cone rod dystrophy, and choroideremia can start in early childhood. Now, inherited retinal dystrophies affect thousands of people, and we are continuing to find ever more genes that are linked to retinal disease. Now, the idea of personalized medicine, while always appealing, is now even more difficult because this is going to require targeting and correcting possibly thousands of unique mutations, given the wide amount of heterogeneity that we can see here. So to address this challenge, the National Eye Institute introduced a program about 7 or 8 years ago called the Audacious Goals Initiative. Now, the goal of this effort was to push the boundaries of vision research and find ways to regenerate neurons within the eye and the visual system. Their targets were photoreceptors and ganglion cells, with particular emphasis placed on regenerating photoreceptors. Now, the goal of regenerating photoreceptors in humans emerged from the knowledge that some species already have the ability to repair damaged photoreceptors. Building off initial studies in goldfish from the 1970s and early 1980s, later on in the late 90s and early 2000s, a number of groups began investigating tissue regeneration in the zebrafish. Now, zebrafish are small freshwater animals. They reach about 1 inch to 1.5 inches in size. They're inexpensive to maintain, and our current zebrafish facility at the Lerner Research Institute, as you can see a part of right here, holds several hundred tanks, and we have the capacity for far more than 20,000 zebrafish. Fish eyes have similar anatomy and physiology to humans, and more than 70% of the human genes are also found in fish. Critically, zebrafish have the capacity to regenerate damaged tissue in the retina, the brain, the spinal cord, the heart, pancreas, and even appendages such as the fins. When you damage photoreceptors, so here we have a small schematic of rod and cone photoreceptors here. If you damage these photoreceptors with high intensity light or with mechanical injury or with chemical injury, they release a number of molecules, and these molecules will trigger another cell type in the retina called the Mueller glial cell to begin to divide. And these Mueller glial cells will form a type of stem cell. These stem cells will continue to divide, and eventually they will mature and regenerate and restore the lost rods and the cones. Now, we've learned a great deal about this process over the last 20 years, but much of our knowledge comes from the studies where the injury is, where the retina is injured acutely by high intensity light damage or mechanical injury, not necessarily through genetic disorders. Here's an example of what how we monitor this type of regeneration in the laboratory. So what you see on top are basically sections of a zebrafish retina that's been stained with markers to identify the cones. The red marker here marks the outer segments of the cones, while the green marker here labels the inner segments and the cell bodies of the cones. The bottom panels show retinas stained with a marker to label proliferating cells, or cells that are undergoing active mitosis and are dividing. Here on the left, when the retina is uninjured, you can see that the cones are aligned very nicely and there's no proliferation, no cell division going on. If we injure this retina with high intensity light damage, you can see that the cones become disorganized. There is even a region where the photoreceptors have been completely ablated, and you see widespread cell division going on within this part of the retina. If you wait for roughly 2 weeks, this retina completely repairs itself, and you can see that these cones have now been regenerated, and there's very little active cell division still ongoing. Now, although regeneration is great following light damage, we want to be able to understand whether or not regeneration occurs in fish with genetic forms of blindness. So to address this question, we've been investigating zebrafish that carry mutations in genes known to cause retinal degeneration in humans. In this case, we're studying a fish with mutations in a gene called SEP290. This gene has been linked to Leber's congenital amaurosis, a disease known as Bardet-Biedl syndrome, as well as retinitis pigmentosa. You can see here, this is the mutant animal. These are normal sibling animals that do not carry the mutations. And you can see the clear difference here. There's an overt spinal curvature. These are micro CT scans showing the scoliosis within the spinal cord. There's fertility issues and a number of other pathologies that we can detect. And importantly, these mutants also undergo photoreceptor degeneration. So I'm showing you again similar images of zebrafish retinas stained with markers for the cone outer segments in red and the inner segments in the cell bodies here in green. You can tell pretty quickly, these are normal animals on top, these are the mutant animals on bottom, that the cones in the mutant animals are both more disorganized as well as fewer in number than what you see from their normal siblings. So this is true both in younger animals at roughly 3 months of age, as well as older animals that are about 6 months of age. Importantly, you can see that the cones do not appear to be regenerating. So have these animals lost the capacity to regenerate? What we did was take both siblings as well as the mutant animals and performed light damage on these fish.
1 minute.
You can see here that the photoreceptors are degenerating, and then they regenerate again after about 2 weeks. As the cones degenerate, we wondered whether the disease environment would limit the ability to fully regenerate. If we light damage a normal retina, the cones are lost, but then they regenerate back to roughly a normal density. In our mutants, when they're already at lower density due to the ongoing degeneration, if we further damage the retina, will they regenerate back to a fully normal, healthy state, or do they only regenerate back to the pre-damaged state? It turns out that when we tracked where the photoreceptors were regenerating, in normal animals, the regeneration would occur to where the cones were back to densities that were almost equivalent to what was happening in the uninjured retina. In our mutants, we found a similar result where the regenerated photoreceptors were at roughly the same density as the uninjured retina, but this was still far less than what was going on in our normal animals. This is critical because it indicates that the disease environment somehow limits the overall capacity for regeneration, and it means that we have a lot of work to do. So our ongoing research questions are, why does the retina respond differently to damage, acute damage versus disease? Why are our mutants not regenerating naturally? Does the disease retina limit the potential for full restoration of the cone densities? We're also exploring what role immune cells play in either promoting or inhibiting this regeneration. And obviously, we want to be able to stimulate regeneration without causing additional damage to the retina. And so translating these findings to mammalian models of blindness will hopefully allow us to develop better therapies. So I'd like to thank many of the people in the lab that have been doing this work, a number of colleagues that have helped out with conversations and reagents, as well as obviously funding. The NIH, RPV, Cleveland Eye Bank Foundation have all been very generous with their support. So thank you for your attention.
Thank you, Brian. We'll move on to our next speaker, Irina Pikoleva, who's a basic scientist at Case Western Reserve Ophthalmology and University Hospital. She's a director of the Visual Sciences Research Center. She's going to be talking about in a search of new therapeutics for age-related macular degeneration. Irina?
Thank you, Dr. Lass. Good afternoon, everyone. So first of all, I would like to thank the Cleveland Eye Bank Foundation for support and for the opportunity to share some of our data with you. Today, I will be focusing on a project supported by the Cleveland Eye Bank Foundation, which is aimed at the search of new therapeutics for age-related macular degeneration. So what is age-related macular degeneration, or AMD? It's a leading cause of blindness in Americans over 60 years of age. It's a progressive degenerative disease which destroys central vision due to changes in the macula. This is a model of human eye, and it shows where macula is located, and this is a brown spot in the back of the eye. And this picture shows how world is perceived by somebody who has problems with the macula. Age-related macular degeneration starts with appearing in the fundus, and this is the fundus photo, of cholesterol-rich pale yellowish deposits called drusen. And as disease progresses, these deposits grow, and then AMD can follow two paths. Either become a dry AMD, or this is a geographic atrophy, or thinning of the retina, or become a wet AMD, which is characterized by abnormal growth of blood vessels in the retina, and these blood vessels are leaky, and therefore there is some blood in the retina. So my lab is focused on cholesterol and strongly supports drug repurposing or use of drugs that already have been approved by the FDA for purpose other than they were originally approved for. And in my talk today, I will focus on 2-hydroxypropyl beta-cyclodextrin, or HPCD, which is a hollow, shaped like a hollow truncated cone. And the inner surface of this cone is lipophilic, or it likes hydrophobic compounds, like many drugs. And the outer surface of this compound is hydrophilic. And what such properties enable HPCD to encapsulate hydrophobic medications and form water-soluble inclusion complexes. Therefore, because of this property, HPCD was approved as a vehicle for drug delivery. But we also know from basic science research that HPCD can lower cholesterol in different tissues, and therefore currently HPCD is tested off-label for cholesterol lowering in 2 clinical trials in children with a genetic disease. So we decided to test HPCD for retinal cholesterol lowering because drusen are full with cholesterol, and we were thinking of whether we can treat at first mice with HPCD and to remove cholesterol from drusen. And for this purpose, we use 2 groups of animals: mice with normal retinal cholesterol and genetically altered mice in which retinal cholesterol is increased. And we addressed 4 questions in our study. The first question was, does HPCD cross the blood-retina barrier? The retina, unlike many other organs, is separated from the systemic circulation by the barrier, meaning that many compounds that are circulating in the blood, including drugs, they cannot cross the blood-retina barrier. The second question was, what is the best way to deliver HPCD to the retina, and what should be the dose? We would like to know, of course, whether HPCD lowers retinal cholesterol, and since we are basic scientists, of course, we are always interested in mechanisms of the drug effect. So to address the first question, we used 2 types of HPCD: normal HPCD and HPCD with a fluorescent tag. And we used wild-type mice with normal cholesterol and genetically modified mice or double knockout mice. And we injected HPCD and HPCD with a fluorescent tag into mice. And what we see approximately a half hour after injection in both wild-type and double knockout animals, we see HPCD in retinal blood vessels. And by 2 hours, we see that HPCD diffuses from retinal blood vessels into the retina. And by 8 hours, it is washed out from the retina. So to address our second questions, we tested oral gavage, which is similar to humans taking a pill. And we measured retinal cholesterol in HPCD-treated double knockout mice with increased retinal cholesterol and saw a significant retinal cholesterol lowering. And we also tested normal mice. And we see that even if cholesterol is normal, it still can be reduced by HPCD treatment. Then we decided to test whether we can improve this result. And we tested the subcutaneous, we used subcutaneous injections. We increased the drug dose and we did subcutaneous injections 2 times a week or 3 times a week for 29 days or for 14 days. And again, what you see that even with this route of delivery, we see a decrease in retinal cholesterol in treated versus control animals. But if you compare the extent of the reduction, the extent of reduction was smaller as compared to oral gavage. And to address the mechanisms of HPCD effect, we tested the expression of a number of cholesterol-related genes, and we used some proteomics approaches and measured the protein levels in the retina of HPCD-treated versus vehicle-treated mice. And I will not go into the details of these studies. I will only show you that in both animal models, we obtained evidence indicated that HPCD can be taken up by the cell, get in complex with cholesterol, and then be effluxed from the cell. HPCD can also get to the cell surface and extract cholesterol from plasma membrane. And this will lead to a cholesterol reduction as well.
1 minute.
And collectively, based on our studies, our conclusion that yes, indeed, HPCD should be further considered as a potential therapeutics for AMD. And I would like to acknowledge people in my lab. And first of all, the first author on this paper, Nicole El-Darzi, a talented MD from Lebanon, as well as other lab members. I would like to acknowledge the case co-facilities and of course financial support, because basic science research, especially mouse research, is extremely expensive nowadays. Thank you for your attention.
Okay, if we could also have Brian and Paul, their unmute and their video. Okay, well, we're a little behind schedule. I did get some more technical mechanistic questions, which I'm going to reserve for later on. I'd rather give you some more softball lay kinds of questions in terms of the application to disease. So Paul, you're one of the leaders in structural biology and the retina. And where do you see in terms of drug development, how does structural biology promote a easier pathway for new drug development?
I think structural biology, will be really helpful in small molecule therapeutics, because as we're getting more structural information.
What do you mean small molecule?
Like small compounds, like chemical compounds that, rather than maybe, genetic approaches or things like that. And the reason being, as we get more structural information, we actually know what the proteins look like. We actually know where they're at. And then you could turn to computational methods to kind of computationally model things. And then you could screen drugs computationally rather than sitting on the bench and laboriously doing these type of things. So I think that's where structural biology is really making an impact. And of course, once you get compounds, you of course have to test them, in cells and animal models, and then eventually in patients.
Can you give an example in medicine or in ophthalmology where it's already come to fruition?
I mean, I don't know to fruition, but I mean, one of the major advances over the past decade has been in the structural biology of G protein coupled receptors, which are one of the largest drug targets currently on the market. So I think there's been an explosion of actual pictures of what these proteins look like, and as a result of that, there's been just exponential increase in potential therapeutics that people are coming up with in terms of where they are in terms of clinical trials and that type of stuff. I don't have the information on that, but I mean, over the next few years, you'll definitely be seeing some of the fruits of that actually, making it to market.
Great, thanks. Exciting area. Brian, I learned in med school when you and that was a while ago, that if you damage the brain or the retina, there's no way to, bring it back. And so what is the technology that has allowed the possibility of regeneration, stem cells regeneration? What is happening now that this is now a possibility?
So I think there's a few things. We're learning a lot about the genes that control, both positively and negatively, the ability of these Mueller cells to begin to proliferate. And as we, a lot of that work came out of zebrafish, but as people are now identifying those genes, they're able to then activate those genes in mice, and they can see early stages of the regeneration process ongoing in mammals. I think it's still kind of an open question of, is the mammalian retina, mouse or humans, is it prohibitive for regeneration? Things are, those pathways are turned off or have been lost, or is it maybe the lack of ability to stimulate them properly? So is it the absence of a sufficient trigger, or is it things that are actively suppressing it? And I think that's still kind of a open question, but artificially, I think we're learning more about how to probably trigger these things to kick in, even in the absence of damage. But this is all within the last probably 3 to 4 years that those studies have been started. There's obviously another kind of different approach of using real stem cells and transplanting those into retinas to possibly restore vision as well. And I think that's a sort of a different conversation, but one that's getting a lot of traction.
Thank you. And Irina, you're one of the leaders in terms of cholesterol metabolism and its relation to macular degeneration. The practical question is, should everyone be on a specific diet? What about the use of statins? What should we be doing until your work continues to find new discoveries?
Sure, statins were shown to have positive effects on early stage age-related macular degeneration. The answer is yes, continue to be on a statin. It may help, to prevent progression of AMD. Vavas from Harvard showed this effect of statins. And it was supported also in a clinical trial in Australia. Yes.
All right, let me see if we have any other questions here. Cholesterol is essential for cellular function. What is the side effect of HPCD? Irina?
Okay, in mice, it causes diarrhea. In humans, we don't know yet, because it will be different dosages as compared when it is used as a drug delivery vehicle. So therefore, we don't know what would be the side effects in humans. We know that mice tolerate very well very high doses of HPCD, but develop diarrhea. Humans do not seem to have such an effect.
All right, great. Well, we're going to move on. We hopefully will have a little time at the end here. Thank you, everyone. Our next, we're going to go on to our area of diabetic retinopathy and imaging. And our first speaker in this group will be Dr. Patricia Taylor. She's a basic scientist at Case Western Reserve in ophthalmology and also at the Stokes VA Medical Center. And she's going to talk about inflammation and diabetic retinopathy. Patty?
Hi. All right. So thank you very much for allowing me to speak today. Let me share my screen. Today I will be talking about inflammation and diabetic retinopathy, a little bit about diabetes and diabetic retinopathy. More than 9% of the US population is diabetic, with an extra 86 million Americans currently being treated for pre-diabetes. Almost all type 1 and most type 2 diabetics will develop diabetic retinopathy in their lifetime. Diabetic retinopathy is a diabetes complication where blood vessels at the back of the eye are damaged, and this leads to vision loss. Due to the prevalence of diabetes, diabetic retinopathy is now the leading cause of blindness in the working age population worldwide. So how does inflammation induce diabetic retinopathy? Diabetes mediates a continuous or chronic inflammation. Inflammatory proteins such as cytokines are continuously produced. Chemical signaling does not turn off. The chronic response can become toxic to the body and induce vascular damage that leads to retinopathy. One of the most prevalent inflammatory cytokines detected in the blood of diabetics is IL-17A. In healthy individuals, IL-17A is induced for a short period during infection and injury and then shut off. However, in diabetics, IL-17A is continuously produced. Previous studies of other autoimmune diseases have provided evidence that continual production of IL-17A induces tissue and vascular damage. In our studies, we wanted to determine if IL-17A played a role in the onset of diabetic retinopathy. In a pilot study of humans with type 1 diabetes, we drew the blood and collected the plasma of peripheral blood from non-diabetics, diabetics without diabetic retinopathy, and diabetics with diabetic retinopathy. And in this small pilot study of 4 people per group, we quantified IL-17A in the plasma by ELISA. And as you can see, there's basically no IL-17A detected in the blood of non-diabetics. There is IL-17A detected in the blood of all diabetics, but when someone has diabetic retinopathy, it's significantly higher than in the diabetics without diabetic retinopathy. We further looked at vitreous that was collected from cadaver eyes in both non-diabetics and diabetics with diabetic retinopathy. Here we had 4 samples of each. This again was just a pilot study, and we did not detect IL-17A in the vitreous of the non-diabetics whatsoever, and we did detect varying levels of IL-17A in the 4 different samples of the vitreous of the cadaver eyes by ELISA detection. To really look at the mechanism and to see if there's really onset with IL-17A and diabetic retinopathy, you have to go into animal modeling. Our animal model that we used was a streptozotocin-induced diabetes model. Here, we use mice. We can use all different types of transgenic mice since it's an inducible model. Mice were injected intraperitoneal for 5 days with streptozotocin, and then we looked at them 17 days later after the last injection of streptozotocin to confirm diabetes. What occurs over those 17 days is that streptozotocin binds to GLUT2, which is a transporter, and this transports streptozotocin into the pancreatic beta cell. Their streptozotocin inhibits glucose metabolism, it damages the DNA of the beta cell, and it inhibits insulin release, basically making the mouse a type 1 diabetic within 22 days from the first injection. And then 2 months after the mouse is confirmed diabetic, we can look at retinal inflammation, oxidative stress, cytokine production, all in the retina. And I won't be showing it, but in our 2-month model, we confirmed that IL-17A was detected in the retina of diabetic but not diabetic mice. 8 months after diabetes is confirmed, the mice develop early stage retinopathy. And we use the hallmarks of what is non-proliferative diabetic retinopathy being vascular permeability, vascular leakage, and capillary degeneration. So here is an example of what we found in the 8-month model. diabetic mice. And we looked at non-diabetic and diabetic wild type mice, which were our C57 black 6. And we looked at non-diabetic and diabetic IL-17A transgenic knockout mice. And like I said, 8 months after diabetes was confirmed, mice were intravenously injected with a FITC BSA fluorescent green fluid, which then we allowed sit for 30 minutes in it, and it went throughout the whole vasculature of the mouse. Retinas of the wild type C57 black 6 and IL-17 knockout mice were collected. They were fixed, and histological retinal cross sections were placed on slides for microscopic analysis. And what we basically looked for here was, as I have highlighted, these green sort of blurry areas, which were the vascular leakage, the FITC leaking out of the retinal vasculature. And we found that there was retinal vascular leakage in the internuclear layer, slightly at the edge of the outer plexiform layer and the outer nuclear layer. And this is a representative picture of a diabetic mouse, wild type. And as you can see in the diabetic IL-17A knockout mice, we really did not see any vascular leakage. We had looked at 10 different mice in each group and then quantified it. And through basic fluorescent units, we found that there was a significant increase in vascular leakage in the diabetic wild type mice when compared to the non-diabetic wild type. And when IL-17A was systemically ablated from these diabetic mice, they actually went down to no leakage as a non-diabetic mouse. So IL-17 was highly dependent component of vascular leakage in these diabetic mice. We further looked at capillary degeneration. Capillary degeneration basically looks at acellular capillaries during the onset of non-proliferative diabetic
1 minute.
retinopathy. You have the death of cells. And as you can see in the wild type diabetic, these are acellular capillaries where the endothelial cells and periocytes have died, and we did not see as much in the IL-17 knockouts. When we quantified capillary degeneration, we found that there was a significant decrease in capillary degeneration when IL-17A was knocked out from the diabetic mouse. So in conclusion, IL-17A was detected in the blood of diabetic patients as a significantly increase in diabetics with diabetic retinopathy. IL-17A is also detected in the vitreous of our cadaver eyes from our pilot study of diabetics with retinopathy. IL-17A in the murine models has been found to induce vascular leakage, capillary degeneration in the retina, and this is considered the onset of diabetic retinopathy or non-proliferative diabetic retinopathy. Thank you very much. I'd like to acknowledge my lab, all of the people in my lab, my collaborating colleague, Tim Kern, the VSRC Corps, and my funding, which is NIH, OLOR, VA, and Ohio Eye Foundation. Thank you.
Thank you, Patty. We'll reserve the questions until the last two speakers. Our next speaker is Dan Martin, who's the chair at the Cleveland Clinic Cole Eye Institute. He's also a professor at the Cleveland Clinic Lerner College of Medicine of Case Western Reserve University. Dan's going to be speaking on top 5 findings from the DRCR Retina Network clinical trials. Dan.
Thank you. Thank you. Oh my. You know what? Can you shoot? You know what? Can you go on to Justice? Justice, are you ready? I'm gonna have to adjust something here.
Sorry. Okay. Our next speaker is Justice Ellers, who's the endowed chair, Harvard Endowed Chair for Ophthalmic Research at the Cleveland Clinic Cole Eye Institute. He's director of the Image Guided Surgery and Advanced Imaging Research there. He's going to be speaking on seeing beyond the images, next generation diagnostic feature assessment and opportunities for personalized medicine. Justice.
Perfect. Thank you so much for the invitation today. Let me just share my screen. I'm going to open up my talk here. Perfect. Is that coming through for everybody?
Looks great.
All right, excellent. Well, thanks again for the introduction, Dr. Lass. It's really a pleasure to be here. I'm going to talk a little bit about some of the exciting areas in imaging and diagnostics in ophthalmology, and potentially some of the opportunities down the road for personalized medicine. These are my financial disclosures. So why do we need precision medicine and image-guided metrics? And you can make a lot of arguments why that might be. When I think about the need, I'd love to be able to optimize diagnosis efficiency. I'd be to more quickly and accurately risk stratify our patients who we see, to determine treatment risk and benefit of whether to actually move forward with a specific therapeutic plan, and to individualized therapeutic approaches. This includes things such as wet macular degeneration, targeting our clinical trial enrollments, particularly as we get more and more therapeutics available, as well as to understand our failures. Why did someone not respond in the way that we wanted to? Improving our economic models, as well as becoming more and more important. So one question is artificial intelligence the answer? Certainly when you read many of the headlines, this is one of the, I think, greatest focuses that we've seen in ophthalmology, and particularly in retina, perhaps since OCT angiography came out. And that is, this ability to have computers help us in the decision-making process, or perhaps even replace us. But even experts in artificial intelligence say that we really should be careful about how we fully trust, some of these brute force algorithms, and to be sure that we understand, what's happening. So where could we use AI? And certainly there's an argument that we could use it everywhere in the way that we take care of patients. But more specifically, I think that there's great opportunity for automated detection of disease, such as normal versus abnormal, perhaps disease characterization, such as severity grading in specific disorders. Big data assimilation is a major opportunity for looking at risk stratification or diagnostic accuracy, as well as enabling next generation disease interrogation, looking at features that we haven't been able to readily use before, and to create opportunities for the future in looking at specific entities. And lastly, to enhance current platforms to improve our efficiency and accuracy of our current workflows. There's also emerging sort of opportunities in pattern recognition and phenotype classification, perhaps even using something such as pharmacogenomics or even imaging genomics to better understand the underlying diseases that we're looking at. So when we think about AI, some things to consider: what's really needed to train the neural network? Well, we need data sets of sufficient size to be available, and we need either annotated or raw images based on the specific features that we're interested in. Another thing for me, as I've learned about this technology, is understanding the thought process. How is the computer actually looking at this? We hear about the black box. Is it really a black box, or is there ways for us to understand what's going on? Because what we really want to also know is what happens when things go wrong with these AI systems. So diabetic retinopathy is really kind of low-hanging fruit and potentially where artificial intelligence could be used. This is a paper here that actually compared a neural network to ophthalmologists, and actually found that the neural network was more accurate compared to multiple different ophthalmologists in detecting the overall levels of diabetic retinopathy. This work and others has led to recent FDA clearance of the first AI-based diagnostic, and that was actually in ophthalmology, specifically looking at diabetic retinopathy. Numerous research groups have looked at this technology and are expanding the role of this automated image analysis and diagnostics. And as we can see here, this is an example of that IDX platform. It's essentially a computer companion for your standard fundus camera. And what this uses is a software assessment to essentially detect what's called more than mild diabetic retinopathy, with an 87% sensitivity and a 90% specificity. And in fact, this system in their work to get this approved identified all patients who had a level of 43 or higher. So again, being sure that we're detecting those people who are at higher risk. So what about detecting systemic features? This was a paper recently that got a lot of press that was done with Google as well as other collaborators. And for us to look at a picture like this to decide whether this individual is male or female, we may feel like there's some contextual features that might point us in a direction. It becomes more difficult with an image like this. But what if I asked you to say, is this a male or a female? Well, although I don't know that a human can do it, a computer can. And what we see here from this paper is that using a very large data set of fundus photos tied with different demographic data, the computer in this AI neural network was able to look at multiple features, including age, gender, and blood pressure. And some of the things in sort of unpacking this black box we can do are these attention maps of where is the neural network looking in these images to make these decisions. And interesting, we can see here the green overlay on the fundus photo is where the greatest focus is for determining the specific question. Ones that I find interesting, for example, in gender, we see that the fovea is actually lighting up. And is there something around the pigment, characteristics within that image that sort of tips off the computer that this is a male or a female? More intuitive, though, is when we look to the right here at the systolic blood pressure measurement, we see that it's really the vessels that are being identified and looked at carefully by the neural network to determine and sort of guess at what the actual predicted systolic blood pressure is. But then there's other ones like diastolic blood pressure where it's green everywhere. And we really don't know what the primary focus is or how it is that it's making those determinations. Interestingly, we can see that the systolic blood pressure was actually more accurate than the diastolic blood pressure predictions. So I want to talk about how we've been using some of this in our lab for computational image assessment and higher order image characterization. Specifically, we'll start with ultra-wide field angiography. And we found that using angiography for retinal vascular disease, particularly ultra-wide field platforms, is really critical in understanding disease burden. But when we look at our management and we use OCT so regularly to make treatment decisions, for managing diabetic retinopathy, how we quantify ultra-wide field angiography has really been lacking. And so using some of these technologies, we've been able to develop platforms to actually quantify leakage, as we can see here, that strongly correlates with the clinical severity. When we want to look at this from the next generation, how do we actually integrate machine learning? And that is to kind of bring together multiple different aspects of this, being able to identify the region of interest, the blood vessels themselves, even where the fovea is. And as many of you have seen in some of these wide field systems, trying to remove the eyelid from the image as well and having an eyelid detector. So just to show you some of the examples of how these neural networks work, we see here, this is our optic nerve detection platform. Sorry, let me go back here. And we see the grayscale image of the confidence of where the machine learning is looking on the bottom row, as the fundus angiography image on the top. And we see here where overall it's quite confident of where the optic nerve actually is. In the third column here, we see a faint area of gray corresponding to this neovascularization that the computer is at least considering. Maybe that's the optic nerve, but ultimately it makes the decision on where that nerve is here.
Sorry, less than 1 minute.
Yep. And so this is the eyelid removal tool here, as well as our leakage detection that we can see over time that we can actually look at this. When we look at feature assessment, we can use artificial intelligence as well for blood vessel segmentation. And using that, we can actually enhance the details. As we can see here, this is a normal eye, almost looks like an OCT angiogram, an eye with proliferative diabetic retinopathy with dramatic dropout, as well as sickle cell retinopathy here with peripheral vascular loss. The last thing I want to just talk about is OCT and how we can use this to look at outer retinal integrity in new ways using these type of networks. Hydroxychloroquine is one of those challenging toxicity things that we try to find on the images. And these are one of the struggles that physicians have in being able to identify subtle changes. What we've been able to do is utilize this technology to see these visualization maps. And what we can see here is much earlier detection of signal of retinopathy that may have applications in the future. The last thing is just to talk about pathology that we can see in wet macular degeneration, being able to quantify things such as fluid and the therapeutic response that we see, and whether that's using traditional image analysis platforms or machine learning enhancements. And as we can see here, we can actually now quantify the volumetric characteristics of fluid, intraretinal fluid and subretinal fluid. And in ways not previously able to be done, we see that sort of three-dimensional aspect of where the fluid is and how it's responded to treatment over time. Just in the interest of time, I'm going to skip forward to my conclusions. And really what's left with this and how do we move forward? Well, AI-based platforms and computational image analysis are really likely to play a major role in eye care for the future. Additional research is needed to better understand how do we integrate this into our practices. And the emerging technology related to this field may really be a major inflection for our field, and how we can really leverage it for personalized care and improvement in overall disease management will be critical for going forward. Again, I want to thank you for the opportunity to be here today and acknowledge my team here at the Cleveland Clinic and my partners at Case Western. Thank you.
Thank you, Justice. Okay, I think Dan is back in shape here. And as again, we're going to have him speak on the top findings from the DRCR Retina Network clinical trials. Dan? And I think you have to go on, you're muted.
Yeah, okay. Yep.
There you go.
My apologies for the glitch. Share my screen. There we go. Yeah, the computer I was using worked perfectly fine earlier this afternoon, sharing screen, but for some reason it wasn't going to do it. So good afternoon, and thank you to the Cleveland Eye Bank Foundation and John and everyone else who helped organize this. I'm going to speak to the top 5 findings from DRCR Retina Network clinical trials. I have no financial interest to disclose. So in 2002, the National Eye Institute sponsored the creation of the Diabetic Retinopathy Clinical Research Network. That's where the DRCR comes from. And for the next 15 years, the network conducted a lot of trials in diabetic retinopathy, examining new treatments, laser photocoagulation, and a variety of different mechanisms of disease. In 2017, the NEI allowed the DRCR to expand its scope to study all retinal diseases. And in 2019, we renamed ourselves as the DRCR Retina Network. For the last 17 years, we've now conducted 30 diabetic retinopathy multi-center clinical trials. We have over 100 publications, 160 clinical sites. It's a very collaborative network. I have the good fortune of serving as one of the 2 national network chairs, along with Jenny Sun at the Joslin Diabetes Center at Harvard. We have over 1,800 members of the network, 505 current investigators. This is a list of the clinical trials that we've completed over the last 17 years. And I'm going to tell you about some of the ones that we're about to report out and some ongoing trials some trials that we're currently planning. But first, I'm going to tell you a little bit about diabetic retinopathy. So if we could go down to the surface of the retina and look at the macula in profile, this is what it looks like. When you image it with OCT, this is what it looks like. This is the image that we capture every day in clinic, similar to some of the images that Justice just showed you. In diabetic retinopathy, you have progressive damage to the endothelium, and you have the formation of microaneurysms, leakage of fluid into the retina with exudated precipitates in the retina, and collection of fluid in the macula that results in loss of vision. That loss of vision has been the subject of many, many clinical trials. So I'm going to start out telling you about a few of these. These are really seminal findings when we first reported them. The first is that anti-VEGF agents are the most effective monotherapy for diabetic macular edema. This was DRCR protocol I. Nowadays, this seems like old news, but there's an enormous amount of information that we continue to extract from this protocol. It was 854 eyes with diabetic macular edema, vision between 2032 and 2320, randomly assigned to laser, ranibizumab, an anti-VEGF agent, a combination of laser plus ranibizumab, or triamcinolone. The primary outcome was at 1 year. What we basically showed was that compared to laser treatment, there was a significant gain in vision. This large difference seen here was accomplished with the use of either ranibizumab alone or ranibizumab plus laser. For the most part, we actually rarely use laser now for diabetic macular edema. The second finding from that study, and number 2 for this series, that the need for these injections decreases over a 5-year period. To me, this is one of the most remarkable and newest findings, something we did not know. I can remember treating patients with diabetic macular edema with laser photocoagulation over and over and over, and eventually it would sort of begin to get better. I now recognize that some of that might have been natural history. And then certainly with anti-VEGF treatment, we now know that if you treat patients intensively during the first year, typically in the year 2, the number of injections declines. You can see the mean numbers 8 in year 1, 2 in year 2, and 3 and 1 in year 3. In other words, diabetic macular edema with appropriate treatment can be a relatively self-limited disease. That was completely unknown prior to this observation. This was the first study that showed that. There were multiple protocols that have been conducted since then that have replicated this finding. Number 3, not all anti-VEGF agents are equal for diabetic macular edema. This was DRCR Protocol T, which we published in the New England Journal in 2015. We compared head-to-head the 3 dominant anti-VEGF drugs, bevacizumab, ranibizumab, and aflibercept. And what we showed was that for all comers, aflibercept was better at year 1, at 2. But the best way to look at this data was to separate it out in a way that we had pre-specified that we would, to look at it according to visual acuity, and that was people with good vision, 20/30, 2 to 20/40. Sort of didn't matter which drug you used. They were about the same at 1 year, and even at 2 years, they were still about the same. They were actually statistically the same. But for the people who had worse vision to start with, 20/50 or worse, aflibercept clearly did better than the other 2. So as a result of this study, the standard of care now is that when a patient comes in the door with 20/40 or better vision, you can choose whatever drug you want because Avastin is much less expensive, 40 times less expensive than ranibizumab and bevacizumab. And aflibercept, we tend to start with Avastin, or many people do. But if you come in with worse vision, we tend to start with Eylea. You can start with Avastin, and there's another DRCR protocol that's been very interesting that we're currently conducting that examines this question, and that is, while there was a clear improvement with visual acuity with aflibercept in more severely affected eyes, the absolute difference between drugs wasn't that great. Only about a 6% absolute difference of 6% between aflibercept and bevacizumab. In other words, if you started with bevacizumab, you could still accomplish most of the visual acuity improvement. And so there's another protocol we're conducting right now and it's asking that question. If we start with bevacizumab first and we don't accomplish the level of visual acuity improvement we would like, and we switch to aflibercept, are we compromising long-term visual outcome by not starting with aflibercept first?
A little less than 1 minute.
Protocol V looked at eyes with good vision, 20/25 or better. And basically we published this in JAMA just last year. We basically showed that for people with good vision, observation was just as good as immediate treatment, and that over a 2-year period, you did not compromise long-term outcome if you delayed treatment until they absolutely needed it. The vast majority of people were 20/25 or better, no matter how you started. Most people never needed, 2/3 of people never needed an injection. And finally, protocol S, which looked at severe diabetic retinopathy and anti-VEGF therapy for that. This is a fluorescein angiogram of proliferative disease. This is what neovascularization in PDR looks like, with severe vessels due to severe ischemia that leads to bleeding and loss of vision. The old treatment for this was laser photocoagulation. We now treat this frequently with anti-VEGF therapy, and this was the clinical trial that showed that anti-VEGF therapy was equal to PRP, and in fact, in many ways, had some benefits over PRP. So there are disadvantages of not doing PRP. We still do it laser photocoagulation because the injections are expensive and they require people to come in. And by the time you have proliferative disease, sometimes people get sick and they miss their injections. That can result in catastrophic results. These are the ongoing clinical trials that we're completing. We're about to report out. Actually, I have 2 that are not even listed here. We report one at the Academy. We've got another one that we're reporting at the Macular Society. Then we have W is going to report out next year. AC is going to report out, and AE. And then we have a whole host of other clinical trials that are currently in design, looking at fenofibrate for diabetic retinopathy, a 4-year long-term AMD outcomes trial, prevention of radiation retinopathy, vitreoretinal surgery for epiretinal membrane, and we're also doing an ROP study in conjunction with PDIC looking at bevacizumab versus laser photocoagulation for patients, for babies with severe zone 1 ROP. Thank you very much for your attention.
Thanks, Dan. If Justice and Patty could also come off mute in their video. Patty, we have a question for you. Do you see vascular leakage on a fluorescein angiogram in your diabetic mice?
I'm sorry, what were you asking? Like, I still can't get my video working. So anyways, you can just hear me.
All right, fine. Do you see vascular leakage on a fluorescein angiogram in your diabetic mice?
Yes, we do. It's not as extreme as you would see in like a macular degeneration model, but you do see vascular leakage through FA. And we started to see differences between non-diabetics and diabetics around 6 months post-diabetes. So it's a little bit harder to quantify as much as the cross sections, but we do see differences.
Just as I asked Irina, do you see, what do you see in terms of the use of anti-inflammatories, non-steroidals, et cetera, for the management of diabetic retinopathy or its prevention?
Well, so we're starting a clinical study at the VA that'll go for 4 years. And we're hoping to actually look at a role of anti-IL-17A, which is an FDA approved drug being used for psoriasis right now. And we're hoping to see that it will play a role, maybe correlating with anti-VEGF, using both as a treatment or as an alternative for those 20 to 30% that do not respond to anti-VEGF. Anti-IL-17A is being used right now, or it's being studied right now for possible use for renal cancer for patients that are not taking well to anti-VEGF treatment. So that's pretty much what we think.
Dan, is DRCR looking at inflammation all in terms of its management in diabetic retinopathy? Are there any plans for looking at that?
So we are.
With your network?
Yep, we did a steroid trial looking at adjunctive steroids in addition to anti-VEGF therapy that showed that you improved the diabetic macular edema, but actually it wasn't a visual acuity result, a visual acuity improvement long term. There's a couple of other companies that have approached us with some anti-inflammatory approaches that we're exploring right now.
So with all the incredible success of DRCR, what would you advise publicly with the incredible increase in the number of diabetics in the population? What is the best thing we can do in terms of prevention of diabetic retinopathy? It's early stages, it's late stages, et cetera. We've heard a lot about treatment, but in terms of prevention.
$1 million question, and we're for something. So one of the things that a big study we're about to launch, likely the largest study we've ever done, is a fenofibrate trial. There's pretty good evidence from 2 studies of fenofibrate for cardiovascular disease in patients in diabetes, and there's another study that looked at it. It was a secondary analysis. Both of them suggested that fenofibrate was beneficial for preventing progression of diabetic retinopathy. So that's the next sort of big DR study. Beyond that, I wish I had the other than tell people to control their A1C. That the most highly correlated metric with progression of or development of diabetic retinopathy and progression is the hemoglobin A1C. How well do you control your blood sugar over time?
I have another question for you, Dan. Is the percent of non-responders known for anti-VEGF in diabetic retinopathy?
Yes, it is. It's very, very well defined. And it's, although it depends on what drug how you define it and what drug you're choosing to look at. One of the really interesting things in protocol U, which was a group of patients who were supposedly diabetic macular edema non-responders, whom had been treated with a minimum number of anti-VEGF injections. Before we brought them into the study, we said, you know what, let's maximally treat these patients. Let's not assume they really have been with anti-VEGF therapy. And it turns out more than a third of the people who we thought were eligible were not, because their diabetic macular edema went away with just anti-VEGF therapy.
Justice, there is now a commercially available instrument for detecting diabetic retinopathy.
Yeah.
How do you see that being deployed? And then when will we have a machine for macular degeneration, be able to go to Walmart to check out whether we have diabetic retinopathy or macular degeneration, just stick our head in this instrument and know whether we got it or not?
Yeah, you know, so it's interesting because IDX was approved, but because it's not built into the camera system, it is another layer, you know, that you have to have in place.
IDX is the company that, right?
You're correct. And it's essentially a computer add-on. The photos get imported in. I think that we're going to see this more and more over the next few years. Obviously, the bar for the FDA in terms of approval, you see this in the IDX approval, which is interesting, which was more than mild retinopathy. And that's something that we haven't actually used clinically, but it makes a lot of sense when you think about an automated diagnostic. You want to catch those eyes that are more at risk. You know, the mild retinopathy, if they slip through, the overall risk to those patients is probably small. I think that we're going to see a proliferation of this. There's companies that are developing home OCT systems that have, you know, that will likely have automated software for detecting disease activity such as intraretinal fluid and subretinal fluid. And it wouldn't be surprising to see something at a Walmart or a Target like the blood pressure cuffs, where you can go in and it's a click fee and you pay your $50 and you get your scan done. I think that that hasn't been the path that most companies are taking right now, but I think there's going to be more and more telemedicine applications, you know, whether it's remote home monitoring, you know, or being done in a primary care office. You know, it's just how do we handle the logistics? That's, I think, the hardest thing in uptake is having the photographers to take it and use the software and all that when you're in a non-ophthalmic office.
Yeah, I mean, with an aging population, we're going to need other ways of screening for these diseases affecting the older population, and so these are clearly not out of the question. So keep checking on what's going on at Walmart there.
Yeah.
So
you know, and I think with that also
And
risk
CBS
stratification, you know, will be important too, of knowing who should we be treating and who should we not.
All right. I think we just have a few more minutes. I don't know if the panelists have any other questions of the other panelists. If they want to come off mute and everybody with their video, maybe someone would take a picture of us, all of us here. If everyone could come off mute and their video. All right. Excellent. Very good. Any other questions for anybody? Because we don't have really anything else from the audience. No, the other ones are more technical. Okay, well, I will thank all the panelists for what I think a very successful first symposium of the Cleveland Eye Bank Foundation. We will be said for all the participants, thank you for also attending our meeting, and we will be sending out our contact information for our speakers in case you have any other questions, want to follow up with any stories that you want to develop. And I'll turn it over to Alan or Debbie.
This is Alan here. Can you hear me?
Yes, very good.
Okay, I can't get my video going right now.
There you go.
Maybe I can. Here we are. Okay, so I want to thank everybody for attending the Cleveland Eye Bank Foundation's Virtual Vision Research Symposium. That was fantastic. We hope you've come away with new knowledge and a sense of excitement of the work and talent that we have in Cleveland and Northeast Ohio. And I would like, and all the impact on the vision that we will have for generations to come. We hope to continue this type of offering and would like your feedback on this first symposium. An evaluation form will be emailed to you in the next several days. A recording of this program can also be found on our website, cleyebankfoundation.org. That's cleyebankfoundation.org. So thank you all.
Thanks.
Thank you. Thank you, John and Alan and Debbie, and really everybody who spoke here. And thank you so much for the support. All of you out there listening, thank you for your support of the Cleveland Eye Bank Foundation. It means a lot to us. It helps us do the work that we do. Thank you.
Thanks.
Thank you.